Hello. Okay, thank you very much for this nice introduction, Shanley. And thanks to EPNOE for this webinar. So the fact that they exist and the fact that they invite me both are great. And so I hope you can hear me correctly. And no, I'm not. And you can see my my screen correctly?
Yes, we hear you and see clearly. Yes.
Okay, perfect. So as you can see, I'm going to present you some research done in my team on nanocelluloses that fix at the oil-water interface. So this is Pickering emulsions. So just to introduce myself, just very fast because Chulin did it already. I am indeed working in the French National Research Institute. That is a community of about 12,000 people all over France. And more precisely, I'm located in Nantes, which is in the west part of France. And we are in a unit that is dedicated to biopolymers, interactions, and assemblies. So about more than 200 people working on these biopolymers mainly. And this is the team I am leading. So it is nanostructured assemblies. And just to give a very short overview of what we are doing. So there is, of course, the Pickering emulsion that I'm going to discuss today, but other kinds, maybe I can, excuse me, the pointer, yes. Then we are working also on programmable materials that can move according to various stimuli that are cellulose based. We made some cholesteric liquid crystal organization based on nanocellulose also. You know that they can make liquid crystals, or we are trying to make nice stimuli responsive materials from hydrogels, foams, or others, or films, for example. So this is a short vision of our work. We are physicochemists working on nanocelluloses. So we are going now to focus on Pickering emulsions. So first, maybe you don't all know what is nanocellulose. So it is obtained from the biomass. All plants are made of cellulose. So it is the first resource from plant biomass. And they exist in the plant as fibers, which are large. But these fibers are themselves made of microfibers, which are themselves made of microfibers. And you can go down to nanometer dimension. So this is a hierarchical structure that makes it very strong. And if we try to recover the smallest parts, then with a mechanical treatment and with enzymatic treatment also, there are what we call the nanofibrils that are quite long, of several micrometers, but still very thin, from 3 to 6 nanometers more or less. And they are very thickening, so it makes very nice gels if you are using it in water. And we can more or less call these spaghetti-like molecules. And then if we compare to cellulose nanocrystals, then it is more rice-like particles. Then these are small rods that are about 180 nanometers and 6 nanometer in cross section that are obtained by acid hydrolysis. So more precisely, from the long NFC, so nanofibrils. They are built of parts that are crystalline domains and parts that are less ordered domains. And if you hydrolyze with sulfuric acid, not too strongly, then you remove selectively the less ordered domains, and you keep only the crystalline parts. But since we are using sulfuric acid, then you have sulfate groups on the surface, and these charge allow to have a good dispersion in water of the nanocrystals that you can see here on electronic microscopy. So from these nanocrystal, there has been a lot of research to describe their morphology and how they behave. So we know that they have different crystalline plans, so which are more or less hydrophilic. There is no real hydrophobic plan, but there are charges that make electrostatic interactions. There are many OH because these are glucoses. So with the OH, you can make hydrogen bonds. And you can have also a van der Waals force that can play a polar phase. Here, the 200 is a less hydrophilic surface. So the thickness is around 6 nanometers when it comes from wood, because you can have many different kinds of nanocrystals. The most common are from wood or from cotton, and these are the one I'm speaking about now. So the width is more or less 6 nanometers, but as we saw that most of the time they are assembled, it goes up to an average of 22 nanometers, which makes an average of a trimer. It means usually we assume that about 3 nanocrystals are linked one near the other, and the length is about 180, as I said. So it's a good source because it is nano. There is a very high specific surface. Because it is a rod, it is very specific properties, which mean that they can align, they can, that there is a dependence on the orientation, which is nice. They are rigid and stiff. There are varieties of interactions, we just saw that. They are available, sustainable, a lot of sources. So we are speaking about wood, but it can be pulp from paper, it can be any waste, any co-product. And then you can have some for low cost compared to many other polymers. And it is colloidally stable in water. So it makes a lot of very interesting things to work with these nanocrystals. So now let's go to our subjects, which are emulsions. You know that you are using emulsions every day. In the environment, you see emulsions even if you don't know. So in food, that's very easy because it's easier to put fat as droplets in a water system than to eat fat directly. In cosmetic, it's the same. There are quite all emulsions. Detergent, because the active products like perfume, fragrance are hydrophobic, so they are most of the time as emulsions, but also in paints, in coatings like bitumes. So everywhere you have emulsions. And these emulsions, okay, this is because it's nice and it's a good friend who did it. So I show you the non-miscible liquids. So here you have oil and water. And when you mix, you make an emulsion. This emulsion, to be stabilized, need to have a molecule at the interface, which is partially hydrophilic, partially hydrophobic. And then you have the surfactants at the interface. This is the most usual way to stabilize emulsions. This is really the basic way to stabilize emulsions. But it's not the only one, because you see here the surfactants, but you can stabilize with polymers that are amphiphilic polymers or proteins, for example. And a third way is to stabilize with colloidal particles. So this is what we call Pickering emulsions, when it is stabilized by a particle, conversely to the two other ways to stabilize. So this is not new, because we are hearing a lot about Pickering emulsions since now more than 20 years. But you can see here that it was Mr. Pickering who wrote a paper in 1907. So this is quite old. And even Mr. Pickering himself said that someone before him, I messed up, yes, already did the same study and found the same thing. And a few years before him, so the discovery was from Ramsden, but still they kept the name of Pickering for this discovery. So what is important to know for Pickering stabilization? So the energy of detachment is proportional to the square of the particle radius. It means that if you have a surfactant, which is a small molecule, then the energy of detachment is very low. So it will absorb and desorb from the interface all the time. So you need to have surfactant in all the bulk. Then when one molecule is removed from the interface, another one will be preserving this interface from coalescence and fusion of the droplets. So here, when you increase the radius, you see that the energy of detachment is largely increased from several orders, you can see, which means that once it is attached, it is very difficult to remove it from the interface, which makes that it is said irreversibly absorbed at the interface. This is the first main difference. And the other one is that it's not a real difference. It is something that you can find with the surfactant also. According to the surface chemistry, so how, if the surface is hydrophilic or hydrophobic, then you will direct to oil in water or water in oil emulsions. It means that if it's hydrophilic, it's going to weight the hydrophilic part. And if it's hydrophobic, it will weight the oil part. And then it will lead to a curvature that will drive the emulsion, the size, the orientation of the emulsion. So only the surface, because what is in the middle is not useful to know if it is hydrophilic or hydrophobic. So the idea to have such particles is to just modifying the surface. You will be able to change the properties of your emulsion. So why are they so interesting? The main thing is that they are highly stable. So they are able to resist to shear, which is very rare with surfactant, and you need to put really a lot of them. You can disperse in a large volume of solvent. So they are absorbed at the interface, making a kind of shell. And then you can use this object, which is a droplet with solid particles around, as a reactor. You can concentrate. It's stable over time, a very, very long time. And you need a very low amount of stabilizer, since once it is at the interface, you don't need any more to keep it stable. So just to show that many different kinds of particles have been identified to be able to stabilize such interfaces. So you see it can be square, it can be any size. And there are very low rods that are identified. And then cellulose nanocrystals are a nice to play with, because it is a rod, then you can orientate, you can do many things with that. But also you can change the aspect ratio and increase the length and see what is happening when you change the size. So we will see that. So what we discovered now more than 10 years ago is that when you mix cellulose nanocrystals with oil, you make sonication. So you should add, that I didn't write, some salt just to prevent repulsion from the charges. Then you have a very nice emulsion that is completely stable, that you can disperse in water, that resists to many things, so shear, concentrating, dispersion, to pH from 1 to 12, to temperature up to 80 degrees. And you need a very low amount, because when it is on the surface of the droplet, then you finished your preparation, you can play with the emulsion. Okay, you have the model, and you have here a real image of nanocrystals at the surface of a droplet. So just to remind you that according to the source, you have very different capacities of producing cellulose nanocrystals. So for example, with cotton, you have these small nanorods that I presented you just before. But if you're using, for example, bacterial cellulose, then they're a bit longer, a bit less than 1 micron long in average. Or if you use, for example, Cladophora, which is an algae, you can have very long nanocrystals that are still very thin, but here they can be around 5 micron long. So this is what we observed, that you can make such emulsions with all the different kinds of nanocrystals, because they are all always organized by the same way, with the same crystalline organization, same crystalline planes, and then they all make very nice stable emulsions. The only very different thing is that when they are very small, they will be covering the whole surface of the droplets individually. When they are a bit longer, they will make some kind of multi-layers. And when they are very long, then they can make interconnected droplets. So then you can choose according to what you want. And you can see that here you will have a very liquid emulsion, whereas here you will have a kind of gel of droplets. So just to understand how it works. So when you put a low concentration of nanocrystals, you have large droplets, and the more nanocrystals and the smaller the diameter, you increase the volume of emulsion. And then the idea was first to understand what was happening here. So you can see that it's quite easy to understand that if you add more nanocrystals, then you can stabilize a larger surface. And then the smaller the droplet, the larger the surface, the interfacial surface. Then you can stabilize smaller droplets when you add more nanocrystals. So this is what is happening. You add more nanocrystals, you have smaller droplets. Since they are hydrophilic, there is a layer of water around each droplet. Then you increase also the volume of emulsion. And to test the stability, we decided to make a soft centrifugation at 4000 G, would be 400. And then we could see that at low concentration, they don't resist to such centrifugation, and above a certain concentration. So you can see here the results of the volume. There is no more emulsion here. There is a phase separation, and then it is very abrupt that you reach about 74%, which is the most compacting you can do from droplets. So here it is. And you see that you have exactly the same volume. So they are completely packed. And what is happening is, so first, in the first part, as I told you, when you add CNCs, then you decrease the drop diameter because you make small droplets and they will coalesce up to the time there is a coverage that is sufficient to be stable. If we increase the concentration of TNC, then still you have this coverage that is always the same, but at the moment there is no more variation of the diameter. So what is occurring when we increase the amount of nanocrystals? Because when we centrifugated, we saw that there is no CNC in the water. So meaning that all the CNCs were continuing to go at the interface, but without reducing the diameter. So therefore, we went to neutron scattering. So I'm going to show you a few techniques that are susceptible to characterize Pickering emulsions in general. So neutron scattering is very nice because you can play with contrast. And here you have 3 phases. So the water in which the emulsion is, the oil, because it's oil in water emulsion, and you have a shell around that is covering the droplets. So playing with contrasts, which means with hydrogenated or deuterated liquids, whatever the oil or the water, you can match exactly to have the same contrast. And then you don't see, if there is no contrast, you don't see anything. So here you don't see the difference between the oil and the water, so it is completely not visible. You just see the shell here. So if you follow the intensity according to the wavelength, wave number in this case, then you see that there is a part where you have a slope of 2, which means that you are watching a 2D film, and this is a surface, and this is the characteristic of just the shell. And if you follow the intensity and you change a little bit, look at ln the IQ square against Q square, then you have H here, which is a thickness that is in the equation. I don't want to bother you with equations, but just to show that playing with this thing, just by the slope, you can recover the thickness of the shell. So this is what we did. And we found a thickness of 7 nanometers. That is exactly the thickness of nanocrystals. So it was clear that we had a monolayer that is directly in contact with the oil. So then it was at this concentration. And what is happening when we continue to add some CNCs without changing the diameter? So we did exactly the same. And what we saw is that still increasing the concentration, we kept a thickness of 7 nanometers, which means that there is still 1 monolayer on the surface of the droplet. So to explain that, it means that this is one big chance to have nanorods, that there is a minimum coverage to be stable, but after they can orientate and concentrate at the surface to be over covered, which means that they are even more stable than the other emulsions that are with spheres, for example, because here they can orientate and really be highly densely covered. And so just as an illustration of what we can do with neutron scattering. So this is the first one we did, matching the oil and the water. But you can also match the nanocrystals with the water or the nanocrystal with the oil. And then you see all the different interfaces. And looking at the slopes, then you can define what is the kind of interface you have. And this is what we did, showing a smooth surface for the oil and a raw surface for the water, which means that the nanocrystals are completely weighted in the aqueous phase, which is another proof of the attachment on the oil and not in the oil. They are completely weighting the water. So we can see, according to the drop diameter, it was surprising to have this stabilization at about 3 to 5 micron in diameter. So can we go down, or is it due to nanocrystals really? Is it a real limit, or can we go lower? So it is generally a question of energy. So most of the time the emulsions are prepared using rotorstator, which works very well. You can use, if you want a little bit more energy, ultrasonication. This is what we are using, just to be sure that we always have exactly the same, because it's more energy than required to make the emulsion. So it is in the lab, at least, a very nice way to have a stable emulsion that are always the same. Or you can use a high pressure homogenizer, which is requiring a higher volume of emulsion. So we don't use it very often. But this is what we tried for once, to try to go down in size. And from the basic emulsions that we had first. So I'm sorry, you have the bars, but I don't know, there is not the value of the bars. So these emulsions are, as you can see here, with a diameter of 5.5 micrometer in average, and they are very nicely monodispersed. And after several passes, so we did really a lot to be sure we couldn't go below in drop diameter, and we reached 330 nanometers average, which means that there are even smaller ones. But then it asks a big question, because I told you that the nanocrystals are about 180, 200 nanometers long. And how can such particles stabilize droplets? Because they are very long compared to the drop dimension. So could they do that or not? Or stay like this? So we visualized using electronic microscopy. So this is MEB and SEM, sorry, scanning electron microscopy. And you can see that even for very small ones, so this is 100, so you have very small one and more or less longer ones, you can stabilize the emulsion with nanocrystals that are really bending on the surface. There, we expected a kind of nest, but no, the crystals are not that thick, that they can bind nicely, and we see them binding on the surface. So this was very surprising, because we had the vision of a solid rod that couldn't bend. And finally, we found a work led by Karim Mazo on modelization. And finally, they tried by a model system to bind the nanocrystals according to the various crystalline planes. And finally, they showed that there was a bending point at large deformation that is different, very different, according to the crystallographic plane. And that's the 100, that is the most hydrophobic plane, was able to bend. So here it is, without accident. So it means that when you try to use the other ones, you see that you have very strong deformation. So it's very difficult to curve the other surfaces, but this one is possible. So the most hydrophobic planes could be directly here, as you see here, in contact with the oil, and then bending up to 60 degrees angle is possible without any degradation of the nanocrystals. So what we did recently, so this is published in this year, in 2023. We wanted to put an interface. So this is oil and this is water. And then Pan Chen and Yu Chen did the work with Yoshi Nishiyama. So Yu and Pan are from China and Yoshi from CERMAV in Grenoble. And we did put some model nanocrystal in the middle of water and wait to see what is happening. So you see in time, first there is the distance is about 15, whatever. And then when we go down, here is a 0. And you see this is the distance of about 5 that we reach in time. So it was clear that the nanocrystal directly decided to go to the oil, and it turned to orientate a specific plane to go and attach spontaneously the oil. And it was the 200 plane, which is the most hydrophobic or the least hydrophilic plane that is in contact with oil. And it was clear that once it is absorbed, there is no desorption. So what we saw also is that it was spontaneous with a negative free energy. It is indeed irreversible, highly stable. And what is interesting is that it is both due to enthalpy and entropy, but it is mainly due to enthalpy, which was a bit surprising to us. But then this was confirming that there is adsorption directly on the oil with the 200 plane, and that this is spontaneous and irreversible. And then I told you that we can play with the lens to see what is happening. So at the beginning, I told you that we can make spaghettis or rice from wooden samples or cotton samples. So here you have in blue the CNC. So as I showed you in the first part, there is coalescence up to a drop dimension that is with the minimum coverage required for stabilization. Then you reach a limit in diameter, and finally you have a densification of the surface because the cellulose nanocrystals will align. And then with NFC tempo, which are much longer, then they will also do the same for the lowest concentration. But when you increase the concentration, they might have contact, and then there is an increase of the diameter. And finally, the diameter, the more NFC you add, and the larger the diameter. And what we could observe is that you have larger aggregates, so larger associated system, but lower drop diameter. So this is a very important point, that if you have emulsions and you just do granulometry, you can make mistakes, because we could think that we have larger droplets, whereas we just have larger aggregates. So it's very important to correlate what you can see with a granulometer with optical microscopy. So this is something to compare, that the longer, and you can have very different ways to have such kind of emulsions. If we continue, I don't know if there are questions or if we wait for the end.
Let's wait to the end. Yes. Yeah.
Okay.
Please continue. Yes.
So now I'm going to show you something we can do, which is changing the surface. For example, we add some tannic acid, which is very well known to have contact with cellulose. And then if you mix cellulose nanocrystals with tannic acid, both in water, you dialyze to remove what is not absorbed. And you have a very nice colloidal suspension that you can dilute in water with droplets after adding the oil. So we generally use hexadecane because it is something that we know we will have always the same, but you can do it with any vegetable oil or any other kind of oil. It works with many different oils. And so you have Pickering emulsions. So you see that here are the different emulsions you have with different concentrations. So if you increase the concentration, then you still increase the volume. So here we have the results, that in blue it's only CNC, and in red CNC and tannic acid. So there is a difference in emulsion volume, slight difference in diameter, but at high concentration you can see that they are exactly the same. So there is a very small difference. This is really due to CNC, because TA we checked doesn't absorb at all. It is not working. And you can see that they are both stabilizing emulsions. So they are very similar. We can say that there is no difference. However, you see the difference in emulsion phase. So here is just a reminder. I didn't explain you. When we make these droplets from electronic microscopy, we are using styrene in water emulsion. We polymerize the styrene to have a polystyrene latex drop that is made at 70 degrees. And then we can image, we can dry and image in air easily. But it is always the question, is that really representative from what you have if you don't put it in water? So we decided to do some environmental SEM to be able to visualize directly in water. So here you have the surface of the CNC, and I'm sorry, it's not nice, but you can see that the nanocrystals are here, but they are not well defined. That you see that there is a kind of swollen monolayer of CNCs, whereas using tannic acid, the surface is very different, and we did it several times to be sure that you don't have the same surface. So there is really a more cohesive surface with tannic acid. So very soft change in surface, because tannic acid is not really a big difference in surface. Even here we can see some difference. So it's also to present the technique. We used it for several different polymers, but I won't present here. Next time, maybe. But this is a nice way to visualize the surface. No, we said no. So now I'm going quite, do I have a lot of time still? Yes?
Yes, and there's time. No worries. Go ahead, please.
Okay. So now I'm going to present you different ways to use such emulsions. You can wonder, what are you doing with that? So, for example, we can make high internal phase emulsions. So that are called HYPE. So the maximum packing of monodispersed spheres is 74%. You can't put more droplets or beads in a volume, because if they all have the same size. So if you go over 74%, then something must happen. So there are various names. So an emulsion is with an internal phase which is below 50%. If you go over 50%, normally it should inverse the emulsion. But if you don't inverse, but just grow the amount of internal phase in the continuous phase, then it is called MIPE, medium internal phase emulsion. And if you go above 74%, then it is high internal phase emulsion, because you must either deform the droplets without coalescing, or change the size of the droplets and have big and small ones and try to put everybody in the same thing. But whatever, they must touch each other. There is no other way. So we did it with cellulose nanocrystals, increasing the oil content. And you can see that from the basic Pickering, the normal ones that are really liquid, if we increase the amount of oil, then we can go above 50%, but still they are round, more or less. We are at the limit, and then it is a soft gel. But if we continue, then we can completely deform the droplets and go to high internal phase emulsion. So this is something really surprising to us, that with very, very little amount of nanocrystals, we can make so resistant solid gel, which is really a kind of solid gel that that will comprise only oil, a lot of oil, up to more than 80% of oil, less than 1% of cellulose, and some water. That's all, nothing more. And it is very stable also. So this is the kind of image you can have, that you have these walls of water plus cellulose nanocrystals that are susceptible to stabilize all this amount of oil around. Another example is that we can go to foams. So, for example, if you prepare from oil and water an emulsion, then this is the emulsion. We don't see that well, but this is the emulsion. Then you centrifugate to keep here the emulsion at the bottom. You remove the water to keep only this, and then you freeze. And after freeze drying, you have a nice foam that is very stable and is completely made of alveolar structures that are decided by the emulsion properties, which means that the size of the droplet, as you have here, will be the size of the alveolar structure. So you can have such foams. And as I told you, that if you increase the oil phase, then you increase the drop diameter, then you can vary by the same way up to HYPEs the alveolar dimension of the foam, just by changing the properties of your emulsion. So it's really resistant, and it means that these only cellulose made forms. There is nothing else, and very few cellulose is enough to make such structures. So this was used in a specific case to make thermal super-insulating materials. So you see that we took nanocelluloses, so it was NFC, the long ones, the spaghettis, mixed with oils, making the emulsion, freeze-drying, and then having this alveolar structure. And you can see that the thermal conductivity of wood or liège, I don't remember, but it's very used for conductivity. Or this is polyurethane or polystyrene, and here air. So you see that our results are better than air. So this is why it is in the super insulating category. And this is only due to the size of the droplets that are able to make some to slow down the diffusion, whatever. So if I can continue, I present you another example, water in water emulsion. So when you mix two solutions of polymers, so mainly with long chains, the most probable result is that you will have a phase separation. They will mix at very low concentration, but very fast they will make two phases. So this is presented here. At low concentration or very low molar masses, they will be miscible. If for any reason they interact one with each other, it will make coacervation. So mainly if you have a positive one, negative one, for example. And otherwise it will be incompatible. And this is the most occurring case. And then if you mix, you will have droplets of a polymer A rich phase and a polymer B rich phase as dispersed. So this can be described in a phase diagram, and you have a tie line that shows the same interfacial tension. But when you go along this tie line, in fact, I do it again. When you mix polymer 1 and polymer 2, then you will have this concentration of polymer 1, this concentration of polymer 2, which means that you have this composition. Sorry. And this composition is made of a volume fraction of phase 1 and a volume fraction of phase 2. And you can only go on these tie lines, which are parallel. And the further you go from the limit of mixing, here there is only one phase, and here you are in the emulsion biphasic phase. Then when you go far from the limits, you increase the interfacial tension. And when you move along the tie line, then you just change the volume fraction without changing the interfacial tension. So it's a very nice game for that you can change the concentration or the fraction and try to identify what is happening in this kind of water and water emulsions. So what is particular to this system is that first, the interfacial tension is very low. So usually it is around 50 millinewton for oil and water emulsions. And for water, it is around 0.5. So it's very difficult to measure. And the interface is very large because you have two phases of water. So there is not a clear limit in between these two, but a thick one. And then you see that it's not possible to put surfactants in such a thick limit. It's very difficult to find polymers that are stable in the interface. So the colloidal particles are really the best way to see that. So this is what we did, mixing PO and dextran. And you can see that changing the volume fraction. So here you have the dextran in green and PO in transparent. You can make PO in dextran or dextran in PO emulsions. And what we saw is that one is unstable. You can make, but it's unstable. And the other one is very stable. So you can stabilize with CNC. Very nice PO in dextran emulsions. And just to show that there was still a problem of creaming, because you always have one polymer that is lighter than the other. So you know that when you add some salt to CNCs, they are going to interact and make some gel. So using light scattering, we found the gelling time, and then we could follow the gelling of the CNCs according to the salt, the ionic strength. So when you put it in water, it is completely well dispersed. Increasing the salt, we saw that there were some aggregation, but that was not completely random, but following something like fractal association or self-similar aggregates. And here, just I show you what you see when you look on a microscope. So this is just a CNC suspension with some NaCl. Yes. So you see that. And you see that the aggregates, self-similar aggregates, are forming directly on the microscope. So it's really nice to see. And now it won't move that much. It's because on the volume it is better organized, so you have a better contrast, but it is the same aggregates that are formed directly when you add some salt. So we use this property to make some emulgel. It means that you have the droplets of water in water, so PO and dextran, and they are stabilized by aggregates of CNC, which makes which prevents the creaming, and then you can have fully stable emulsions. You tell me, Chunlin.
Maybe yeah. Oh, well, time is a little bit approaching to the 1 hour, but yes, please, Simon. That would be great to quickly see the examples. Yes, thank you.
So another example is we mix starch, so dissolved at high temperature and pressure with the Pickering emulsions. So when you mix that and let it dry, for example, in a Perry dish, then it dries and makes a very nice transparent film. So we wanted to be sure that we had nice droplets in it. So we stabilized this time with chitin nanocrystals. I didn't discuss about chitin, but it's working similarly. And you see that you can have the droplets that are really stable in the complete volume of the film. So here you have yes, I have images. So pure starch makes a nice film, and the more oil, more Pickering emulsions in the mixture, and the more droplets, and they were on the confocal characterized, and you see that they are very well dispersed everywhere. So there is no aggregation. It is completely dispersed in starch. And we can go up to 45% in volume, which is really high. And this was done in Nantes. Okay.
Yeah.
I have a last one, but we can skip it.
Yeah, but that would have been better to listen to, but sorry, we have to maybe give the audience time to answer some questions.
Just
I'd like you to wrap up. Yes, please.
just to thank people. A lot of people have been working on these different subjects. So I wanted to thank everyone that you can see here, from Neutron for aqueous immersions, modelization, and all people from the team.